Thank you so much. It's a pleasure to speak at a symposium that's sponsored in part by the Microbiota Vault. I'm a big believer in preserving biodiversity. My wife, Kyria Bounty Mills, runs the FOF yeast collection here at UC Davis, which is soon to be about the third largest yeast collection in the world. And so this is very near and dear to my heart. I'm very thankful for the Microbial Vault existing first and foremost. So today I hope to talk a little bit about how the study of the early life gut microbiota, and particularly bifidobacteria, and understanding their synergism with milk might allow us to identify novel strains and species that could be helpful for human health. Like Jack, I have some disclosures. Early in life, of course, we're born, as I like to say, reasonably sterile. And our gut microbiome comes from outside, but it is guided by milk. And you can't forget that milk is about 200 million years old because it developed with mammals. And so there's a lot of evolutionary process in the shaping of milk. And part of its function is to shape the gut microbiome. So we start with a low diversity gut microbiome early. And then until once we build up of our initial microbiota, but via mostly transfers from our mother and the environment, and that stays at a low diversity, as I'll talk about in a bit, until we wean. And once we wean, the microbiota shifts dramatically. So what does an infant gut microbiome look like? Actually, this is known for a long time. So more than 100 years ago, Tissier, a French pediatrician, looked under a microscope and saw these Y-shaped bacteria. He called them Bacillus bifidus, and that were completely dominated breastfed infant feces. Ernest Morrow also saw similar things and recorded similar things around the same time. But what does an infant microbiome look like today? This is an old but quite useful chart showing the different taxa that emerge in infant gut microbiomes over time, starting off with a very low diversity microbiome at birth. And with breastfeeding, there is a very familiar predominance of bifidobacteriaceae early on, coinciding with breastfeeding. And then, of course, when you get to weaning and other types of weaning foods, then you bring in a variety of other microbiota. And this is the typical pattern you might see in an industrialized population, so from an industrialized country. And we see a very different pattern in a less industrialized country. So in different parts of the world where breastfeeding goes a lot longer, and a variety of other environmental factors promote a much stronger bifidobacterial bloom during breastfeeding. And so we've been studying that and wondering, why is this different from this? What happened in the industrialized world to lose it? So one question is, well, we knew back when Tissier described this, that at least under the microscope, he could tell that the infant feces was full of bifidobacteria. Did they do any other measurements around that time? Well, a couple of years later, they were using pH to measure the fecal pH of breastfed infants. And here you see a typical plot of about 26 infants in a study. And you notice the drop from day 2 to day 10 of about a little over a pH unit. The fecal pH, and they wrote in this paper at the time, this must be because of fermentation of something inside the infant. And it turns out, if you look at a difference between kids in 1926 and kids today, this is a study that occurred in 2018, where they just looked at old data of fecal pH, and they noticed an increase in fecal pH over time in breastfed infants, suggesting that there's something changing in the last 100 years that's causing a rise in fecal pH. Now, one of the rationales for that, of course, is the loss of bifidobacteria, and the concept that bifidobacteria are being lost from an industrial population is not new. It's actually more than 10 years old. This is a wonderful paper by a famous anaerobic microbiologist, Gerald Tannock, who questioned this right at the start. And even before then, certain subspecies like B. longum subspecies infantis were postulated to be being lost from industrial populations. Why would they be lost? certainly you can see some of the many possible reasons why we could lose populations of bacteria in industrialized settings. And I think the duration of breastfeeding, the use of formula, and perhaps the introduction and overuse of antibiotics might be some of the chief causes. But are there specific species missing? Is it just all bifidobacteria? Well, it's specific species, and it's specific species because human milk contains special fiber in the form of human milk oligosaccharides. This is a wide array of oligosaccharides that are produced by mom, make it through the gut all the way down to the colon of infants, and it turns out certain bifidobacteria grow really well on these oligosaccharides. This is about a summary of about 20 or so years of research from both my lab and many other labs around the world listed here, showing that there is a subset of bifidobacterial species that can grow on human milk oligosaccharides. The winner, as it were, or the one that grows the best, always seems to be B. longum subspecies infantis. It can eat every one of these oligosaccharides. B. bifidum can nibble away at every one of these, but leaves many things behind. And a variety of these other species and subspecies can be found in the breastfed infant gut often, but in terms of being able to grow on a larger set of the human milk oligosaccharides, it's about 1 in 10 or 1 in 20 that could actually grow on that, depending on how many isolates you pull out of a baby. Well, it turns out that B. longum subspecies infantis is definitely being lost in the industrialized communities. And this has been shown a couple of different ways. First and foremost, a group demonstrated that it was missing from United States babies. And then Justin Sonnenberg did some wonderful work on populations in Tanzania and compared them to industrialized settings of infant guts, and noticed that B. infantis dominated the babies in Tanzania, but not not so much the babies in industrial settings that were comparators. And a really interesting find that came a couple of years ago in 2021 was B. infantis, we like to call it B. infantis, is actually at a higher level in old order Mennonite communities. So these are communities that live in a very traditionalist way, non-industrialized way in the United States. And there's many communities like this around the world. And it's very interesting that even within the United States, you can have a difference in B. infantis population level, depending on how you live. Well, one of the benefits of identifying such a population like this is if something's missing, maybe you can put it back. And if you put it back, does it help? And this has been shown a couple of different times now for B. infantis. Research from Jeff Gordon's lab has shown that putting B. infantis back in Bangladeshi kids that are at risk of severe acute malnutrition, that this mitigated some of the severe acute malnutrition. There's several papers now that have come out looking at B. infantis used in premature infants as a supplement in premature infants that are fed, of course, breast milk, and it preventing or decreasing incidence of necrotizing enterocolitis. And there's a variety of other, not just B. infantis strains. B. bifidum has been shown to do similar things. So I like to look at the progression of microbial diversity in humans with this wonderful review, pictorial that came from this review in Nature Review recently. And I would point out that I think we have an understanding now of the microbial diversity and what the pioneer species are in the very earliest stages of life to help guide the right functions. And those functions come from, for instance, a bifidobacteria consuming human milk oligosaccharides, producing the acids and setting the stage for the succession of microbial diversity that comes later. But once we understand these things, this is when we can start to put these tools back. And I would argue that we are now in a moment where we are starting, the analytical chemistry of food is starting to change. We certainly know how to measure microbiomes. We're really good at it. And I think Jack and the previous talk gave a wonderful presentation on just the prowess of microbiome analysis. But we're kind of held back in the food realm and the nutrition realm by not understanding the structures of food. And these initiatives, the Periodic Table of Food Initiative, we have a small initiative here, the Davis Food Glycopedia, where we're really trying to understand the glycostructures of food so we can understand how they progress through the gut and they're used for food. I think weaning becomes a tremendous target, because weaning is where we leave the one food we all shared at one point, which is breast milk, and move on to our own environmental foods. And I think we can learn a lot about how to direct the gut microbiome and improve health by studying weaning. So with that, I'll leave some take-home points. Industrialization has led to the loss of taxa, and this has really been a well-documented aspect in breastfed infants with the loss of bifidobacteria and the concomitant loss of HMO fermentation. You can restore that function and by putting the bifido back in. And of course, that argues we have to save the microbiota communities from many places around the world, from non-industrialized populations, so that we can understand how to redirect back to healthy populations. And as I said before, the opportunity around the weaning space, I think, is tremendous. And if I were a young scientist, I think weaning, if you can learn how to control and take weaning in a positive direction, you can imagine many other places in life where you need to wean again. And so I think that there's great opportunities there. And forgive the typo, I just see I have an environmental typo there. With that, I'd like to thank my acknowledgments, and I'd be happy to answer any questions.